My name is Larry Thiel, and I'm Director of the Global Affairs Center here at Shoreline Community College. And we're pleased to see the turnout we have here tonight. This is the first in a series on biotechnology that we're going to have over the next couple, this quarter and in the future, in future quarters. My wife said, you've got to come up with a definition of biotechnology to start this off. So this is what I came up with. Biotechnology, simply put, is the manipulation of biological processes, especially the utilization of microorganisms and even animal models. This came in an email last night from Dr. Ramirez. Rodents, and he'll explain that. The object is to improve people's lives through research and through use in areas such as medicine, agriculture, even energy production. Biotechnology plays a part. So the effects of biotechnology are really all around us, in the medicine we take, in the food we eat, even the clothing we wear. It's a big part of our lives, but it's kind of invisible. So we begin to shed some light on that topic tonight by discussing an important area of medical research and therapy. So let's get started. It's my pleasure to introduce Dr. Guy Hamilton, Professor of Biology and Director of the Biotechnology Program here at the college, and he's going to welcome our guest of honor. I asked Dr. Hamilton up front to say a couple words about the biotechnology program that he directs. So if he doesn't do that, somebody should ask him a question about that. Take it over, Guy.
Wow. Thank you. So as some of you know, we do have a biotechnology program here, and when I say I know some of you know, because the majority of our biotech students in this year's class are in attendance. They just finished their exam. It was a lab exam. So it's great to have them here and the rest of you here as well. And so what I will say about our biotechnology program is it's a 2-year program for students that don't have 4-year degrees and a 1-year for those that already have 4-year degrees. And one of the outcomes for that program is to give students the skills that they need to compete for jobs and work in labs like Dr. Ramirez. And the interesting thing is I met Dr. Ramirez last year, and really the way we got together, and it's and what we've tried to do as a biotech program is reach out and connect to institutes. We have a number of students that work at Fred Hutchinson Cancer Research Center, because I used to work there, and then we have students that work at Seattle Genetics and Presage Biosciences, and we have a number of informal kind of partnerships that we have with them, and most of them through my connections. The reason I met Dr. Ramirez is through one of our students who she had decided last year that she wanted to work at his institution. And so she started contacting him, and then she would come to me and she said, you need to talk to Dr. Ramirez. And she's sitting right in the audience, right behind you right now. That's Naylam. And so that led to now I've met with Dr. Ramirez and his assistant, Sean Popoff, who's now on our board, and we're working out kind of an arrangement to get more of our students working at his institute. So it's really great to have him here, let him see our students, and let you guys be exposed to the type of work he does here. Now, but Larry did give me the full list of your titles and accomplishments, so I have to go through. It's a scale down. It could be larger. Okay, so he's internationally known for his work in neuronal control of breathing. He established the Center for Integrated Brain Research when he joined Seattle Children's Research Institute in 2008. That's CIBR. Do you guys call it CIBR? Okay. In addition to directing the CIBR, Dr. Ramirez is a professor for neurological surgery and pediatrics and adjunct professor for physiology and biophysics at the University of Washington. He also serves as the associate director for the Center on Health Development and Disability at the University of Washington and the co-director for Neuronal Developmental Research Consortium. There are acronyms for those as well that I'm skipping. The CIBR investigates neuronal mechanisms that lead to a variety of neurological disorders, which provides the foundation for potential cures for breathing disorders and Rett syndrome, sudden infant death syndrome, epilepsy, and other neurological disorders. He joins us tonight, which I hope we can all appreciate hearing all these titles, that we were pretty lucky to get him in. And he just flew in from Peru and is flying out tomorrow morning to Australia. So it's great to have you here and to tell us about your work on some of these disorders. So thank you so much.
And thank you so much for initiating the process. So, yeah, it's a great pleasure here, and I love also interacting with students because at the moment I'm in a research institute and there are not many students' classes that I attend anymore, whereas I came from University of Chicago where I was teaching every year a lot of medical students and undergraduates, and I miss that. So it's good to be here, back in a college. And so what I want to talk today is about the new technologies in brain research. And the title was really given to me. And I thought I want to highlight two things. And one is the opportunities of technologies, but also be totally honest with you and discuss the challenges. You know, what are the problems that we're facing in brain research in order to make really a difference to human life? And in this discussion, I want to start with a major difference between therapy and cure. And it might, you know, for someone not familiar with this, might be the similar thing, but it's really a difference. And I want to start with an example, which is epilepsy. And what you see here is electrocardiograms from the brain of a child. And you can see waves here, and they're kind of rhythmic. And in fact, the whole brain is rhythmic. So while you're listening to me, your brain is 30 hertz rhythmically active. And in fact, the rhythm is not synchronized. So some areas are doing this, others are that, and that's pretty good. But what you see here is something starting that actually is developing into something which is not normal, because now you can see these waves, and this is shown here in more detail, these waves that are very synchronized and very much in sync together, and this is a seizure. And this seizure, you can see, spreads from one spot of the brain to all other spots. And that's when you get, you know, like these movements, or you get blackouts and everything. And this is a phenomenon that is actually not very rare. So 50 million patients worldwide are affected from this. And while I was traveling in Peru, I saw so many people, like relatives from me, that had epilepsy, and they don't have access to the same treatment that we have here. So it's really a very desperate situation. And in the US, we have 2.3 million people with epilepsy. And just in Washington State, we have 15,000 children that are suffering from epilepsy. So it's a very common problem. And you can compare it, and this is really done in the airplane while I flew here. This is comparing to a situation where a room in a house is too hot, okay? Your brain is too hot. It doesn't take much, and then it goes into this oscillation, and it burns a lot of energy, and this is kind of epilepsy as a picture. And what you do right now is to treat the epilepsy is you open kind of a window. You let kind of this hot air out, and you do this by having anti-epileptic drugs that decrease the excitability in the brain. And if you're lucky, your anti-epileptic drug really helps and your brain cools down. It's not going into these oscillations all the time. But unfortunately, in many cases, this room kind of gets hot again after a while. And now you have one more chance to open another window. But if this doesn't help, if you don't get a second drug that helps, the likelihood that the third drug will help is very unlikely. And the epilepsy becomes intractable. And the reason the epilepsy becomes intractable is that you really haven't cured the epilepsy. You have just opened the window, you have just cooled it down, so to speak, but you haven't solved the problem that caused the epilepsy, because inside there could be an oven that is way too hot. There could be a thermostat inside that is set too hot. And so you can open the windows, but you haven't really cured the epilepsy, the cause. And this is really the holy grail for all of us, is to develop a cure. And in order to develop a cure, we need to understand the cause that causes epilepsy. And there are many, many causes that can cause epilepsy. And I will come to this more in more detail. So if we understand the cause, and this is our philosophy, if we understand the cause, the mechanism, then we'll be able to cure. And that's the task that we have here at Seattle Children's Research Institute. And we're sitting here in 1900 and Stewart, which is downtown. And this entire 10th floor is just dedicated to understanding cures, to understanding the brain. And this was the center in April 2009. So we were just setting up, and it's like 30,000 square foot of research space. And when you come now, it's totally full. And we recruited scientists from all over the world and the country. And there are many like postdoctoral researchers, there are clinicians, there are basic scientists. It's a whole mix of people that are all dedicated to find cures and understand the brain. And these are just some of these principal investigators in the center. And what they do is they investigate genes that are related in the brain. They want to understand how genes change the molecules in a cell. They want to understand how cells change in response to genetic changes. They want to understand how networks or how cells together interact to form networks. And they want to understand basically how different networks in the different areas of the brain work together to cause a behavior, in this case, let's say an epileptic seizure, okay? But we use for this a lot of technologies. We use whole brain imaging, we use behavioral models, we use bioengineering, so we make special electrodes that can be implanted in the brain. We study, for example, Parkinson patients that have implanted electrodes by developing new electrodes that are better and more lasting. We use electrophysiology. I will talk about this later on. We use something called optogenetics. It's a very modern tool which I think will be revolutionizing brain research. We use microscopy, we use viral vectors, and we use viral vectors because we can implant genes into the brain that can then change and hopefully cure disorders. We use transgenic approaches. That means we make mice that have the same problems that a human has, so we can study the human disease in the mouse. And we use sequencing to characterize disorders in the brain. And we use all this technology to study many different disorders. We look at autism, we look at autonomic disorders, we look at brain tumors, eating disorders, epilepsy, hydrocephalus, and also impulse disorder. And I want to touch some of those disorders and our approaches to solve those in a minute. And our vision is that we want to understand all the levels of integration. We want to understand how a gene causes the disease, how a gene dysregulates cells, how a gene dysregulates networks, and then leads to a certain neurological disorder. But we want to also understand how the environment affects all this, because, as I will tell you later on, environmental influences play a big role in neural diseases. And the idea is that if we understand these mechanisms, we can cure disorders. And again, I want to give you one example here, and again, I want to go back to epilepsy. And this is here a brain of a patient, is a child that has epilepsy. And this is exactly the trace that I just showed you. You can see these recordings here. These are all electrodes that you put on the brain, that a neurosurgeon puts on the brain if you have a child that is intractable, that doesn't respond to anti-epileptic drugs. So what you do is you open the skull, you put lots of electrodes in, and then you record. And then you wait up to 2 weeks and wait for a seizure to occur. And when this child then develops a seizure, you can say, aha, here the seizure starts and spreads out. And this seizure is happening exactly in this brain region. And what the neurosurgeons are doing now is they take out this piece of tissue, and in many cases, this is causing the problem, and then the child is okay again. And so the idea is that you remove the focus, the seizure focus, but traditionally what you do is you throw the tissue away or you do some histological changes. But what we developed is a mechanism where we go as researchers in the OR, and then we obtain this tissue. So this is this piece of tissue that was taken out from one of those patients, and we take it right away as soon as it comes out of the brain, and we bubble it with oxygen. And then we have to prepare it. We have to understand where is this brain tissue located, what's up, what's down. And then we do the next step. We glue this piece of tissue onto something called an agar block. We use crazy glue like this, and then we put this piece of tissue into a so-called vibratome, and this vibratome can produce very, very thin slices of like 200 micrometer thickness. And then once, so you can see this razor blade comes in. By the way, it looks really like a razor, but it's like a $10,000 up to $14,000 machine here. And you see it cuts into this brain tissue, and it you isolate this piece of tissue, and then what you do is you can store this in these vials, and the tissue will be alive for like at least 24 hours. And now we can use this tissue to investigate. And what we do is we take this tissue and put it in another dish and then impale small electrodes into single nerve cells in the cortex or in the brain. Okay, so this is like 20 micromolar, and that's what we target with electrodes. And then once we record from this, we can mimic a seizure, but now in a single nerve cell, and now we can ask the question, what is actually causing the epilepsy? And again, because the talk is about about technology, you know, this is the machinery that you need for this. Okay, so you see, this was like a Christmas Eve. My wife wasn't too happy, but 2 o'clock at night. But we had this tissue, okay, and then we were working 24 hours, and then you can see all this machinery you need to impale an electrode into these nerve cells and then characterize this. And what you saw was one of those setups. Normally we have like 6 people around the clock trying to understand the disorder. And I want to tell you one story. This is a child. She was like 4 years old back then, and she was totally healthy, no problem, went swimming, got a meningitis, got a virus infection, and then started to get these seizures. So this is this girl developing these seizures, and she had so many seizures that it happened in August. By September, she was already mentally highly handicapped and couldn't go to kindergarten and anything. And so no medication helped, nothing helped. And then we went in and we isolated this tissue from this child. And then you can see here is the seizure that we now replicated in a single cell. And we were able now to test a lot of different drugs on these nerve cells, and found one drug that actually stopped the seizure. And this was really just from this child. So we knew, okay, in this child, this drug will help. And then we went back to the clinician, and they used the drug, and the seizure was gone. Okay, so that was really amazing. I mean, these parents were like totally in heaven again, because, you know, this child had every night like tons of seizures, and there was not a good future ahead of it. But I told you that I want to talk about opportunity, but also the challenges, okay? And what we realized is that this great success was just the beginning of a long journey to develop new therapies and cures for neurological disorders. And because what happened is, 5 years later, unfortunately, the girl developed seizures again. So the drug that we found here was not helping anymore, and the child was back in the hospital. And for us as scientists, it was extremely depressing because, you know, when we were able to heal the seizure, so to speak, it went into 150 TV stations, and we were like, oh my God, you know, we're really great researchers, we can help children. And then when this happened, I tell you, everybody in the lab got in a deep depression, because we felt like we cheated the child. We gave 5 more years, but not helping. At the same time, however, it opened our eye that really we have to rethink epilepsy research. It's not about just controlling the excitability. It's about controlling the cause that caused the seizure of this child. And we went back to look at the histology of these children. And what we noted is that a lot of those epilepsies had nerve cells that were very big. So they were like huge blown up cells in this tissue of these children. And this was an interesting finding, but we didn't know exactly what to do with this. But then I moved to Seattle, and we started to collaborate, and he moved also from Chicago to Seattle. This is Dr. Bill Dobbins and Kathleen Millen. And Dr. Bill Dobbins started brain malformations for 20, 30 years. And he knew exactly, what causes brains to become too big or too small. And he discovered, also together with Kathy, the genes, the pathways that underlie the cell growth in the brain. And so he came up with the idea, together with Kathy Millen, that by acting on this pathway, we can stop the cell growth. And one second, I went back. And what Kathy Millen now did is she mimicked the disorder by making a mouse model where you had also this overgrowth, and they also caused epilepsy. So now we had a mouse model that was behaving exactly like these children. It did not respond to any drug. They had epilepsies. And we used a drug that acted on this pathway that they predicted, and exposure of 1 hour totally wiped out the seizures. So that was the first indication that, in fact, we can cure epilepsy in this particular case. And of course, there are many opportunities now, because there are many pathways that we know that can be now targeted. And so we are very hopeful that we can develop actually some cures for these epilepsies. Now, what this showed is that our vision works. Okay, so if you understand the mechanism, you can develop cure. And it also showed that it doesn't take only one researcher, it takes a team. You have to have a specialist for human genetics, you have a specialist for mouse genetics, you have to have a clinician, you have to have these network people in order to actually unravel this. And however, it also told us a lot of challenges. And one of the challenges is, yes, we have a lot of opportunities, but many of these pathways are actually also bad for the brain. Okay, so you don't want to stop cell growth, so you can use anti-cancer drugs, but maybe it stops the seizure, but we don't know exactly what it does to the rest of the brain or to the rest of the body. So what we need to develop is targeting these pathways in very specific areas of the brain, and that's kind of one of the challenges that we're facing. But really, the other major challenge we're facing is that epilepsy is just the tip of the iceberg. And myself as a director, I get all the time, these emails from parents that have a child with a lot of neurological disorders, and they always tell us, these diseases are no problem, but this disease is the biggest problem. So there's a lot of demand for better understanding. And so I talked about seizures, but the reality is this. We have a lot of autoimmune diseases like multiple sclerosis affects 350,000. There's a lot of children with mitochondrial diseases, which also affects the brain very much. There are many brain tumors, 200,000 per year, and these are some of the most deadliest things you can have. Hydrocephalus affects 1 in 1,000 births. So we have a lot of children with hydrocephalus that are born, and they have cerebral palsy, and they have to have shunts, and each time they go to the neurosurgeon to implant these shunts, they're in danger to get a brain infection and come out very badly. Then a lot of children have respiratory disorders. We have still 6,000 children that die of sudden infant death syndrome per year. And then a lot of patients have sleep apnea, 12 million. And that has also a lot of bad consequences. And the increase in sleep apnea is related to obesity. So we have 8 in 10 over 25. So 8 in 10 people over 25 are overweight. And you might think, okay, this is really an endocrine problem, but it's also a brain problem, because we lost the control to know exactly when we ate enough and when we didn't. Or we lost the control of what to eat and what not to eat. Okay, so therefore it's also a neurological disorder. And then there are many, many psychiatric disorders like autism, 1 in 150 children, and I'm sure it's now 1 in 120 by now. which was like almost a 500% cumulative growth from 1902 to 2003. We have a lot of children with anxiety disorder, bipolar, major depression. So there is a huge amount of disorders, and in total, 1 billion worldwide are affected by some sort of a neurological disorder. And the other major problem that we're facing is the complexity of these disorders. And I told you about Tavian, that had intractable epilepsy because of too big cells in the neocortex. But these are other children. These two children also have epilepsy. And this child has Rett syndrome, this child has Joubert syndrome. They both have not only epilepsy, they have also breathing disorders. They cannot walk, they cannot speak. And yet, the mechanism that caused epilepsy is very different. So if we want to treat the epilepsy or cure the epilepsy of this child, we must develop a totally different treatment than the epilepsies for this child. So all these different epilepsies require different therapies, different cures. And the other problem we're facing is, I'm sorry that I really come up with all the challenges, but I think it's very important to be realistic here. The other problem is that the treatment of most neurological disorders at the moment is unspecific. So we give an anti-epileptic drug, but the person will be tired, might actually get depression because it has other effects. And the major reason is that you affect one pathway, but that pathway is involved in many other things. So that's why you have all these side effects. And because of this, many patients don't take their medication, even though it would help them, and because they don't want to deal with the side effects. And so we have approximately 125,000 people that die, and they shouldn't have died because we have a medication, but they don't want to take it because it has all the side effects. And then we have a huge number of morbidities and mortalities because of medication that help, but have other effects that we don't want to have. And then another big problem we have is, where do we draw the line? You know, like if you have a child that misbehaves in the class, at what point do you start medicating that child? You know, like clearly if this child is becoming a drug addict, becoming involved in criminal activities, this could ruin this child. But sometimes these children are just like wild, have a bad face, and they could turn into an Einstein. And so at what point do you start medication? Again, a huge problem. And this problem, again, is very similar to what the problem is with epilepsy. You know, if you can have a child with attention deficit, you might find a drug that actually controls your attention, and the attention is gone. But the problem is it's not curing the attention problem. It also affects creativity, curiosity, risk-taking. So now you give this drug to a child, and you lose a lot of unwanted things, but also a lot of wanted things. So we have to come up with much more specific substances to really treat these neurological disorders. And so the question is, why is it such a hard problem? Okay, why is the brain such a hard problem? And the reason the brain is a hard problem is because the brain is an organ that is fundamentally different than any other organ in your body. And what I mean with this is, if you, for example, look at the skin, okay? In the skin, you have approximately 4 or 5 different cell types that behave very similar, and you can take a piece of skin, put it from here to here, and it doesn't matter. It will regrow, and you're fine. If you look at the liver, there are maybe 4 or 5 different liver cells, and once you understand how these liver cells work, you understand how the liver works, and you can find a drug against these liver cells. In the brain, however, it fundamentally matters what every single cell is doing, where a cell is located in the brain, and how they talk to each other. For example, if I want to lift my arm, I have to activate very specific nerve cells in my motor cortex. I have to activate very specific nerve cells in my spinal cord. I have to activate very specific nerve cells in the cerebellum. I have to make the plan to move my hand. So it's a very specific act. So I cannot take an area from the brain, put it somewhere else, and it's fine. So we have to, if we want to control something with the arm, we have to go very, very specific. And that's the biggest problem. And this here illustrates this a little bit. So this is just a small snapshot of the hippocampus. And the hippocampus is involved in learning and memory. And so each one of those cells is involved in different types of memories. And so if you want to get rid of nightmares, maybe you have to get rid of these cells, but how do you want to target those, whereas these are good memories? You know, so it's a very big problem here, is the diversity. And you have to realize that in the brain we have 10 to the 11 nerve cells. So it's a huge number. So it's 14 times more than the human population. So now imagine the task. You have to understand 14 times the human population. You have to understand exactly what each of those people are doing. Okay? And you have to also understand how they talk to each other, how they interconnect. And that's kind of the target problem that we have. I have to tell you also some good news, though, is, these people are located in very specific areas. So let's say if you have a set of people that are in Bainbridge Island, they wouldn't move, let's say, to Africa or something. So okay, these people in Bainbridge Island, they do exactly that. And then these people in Mercer Island do that. And that way, yes, it's 14 times the human population, but it's not totally hopeless, okay? But it's still specific. Another very important aspect is that there are many, many more nerve cells than genes, because everybody says, okay, if I understand the genes that cause epilepsy, or if I understand the gene that causes ADHD or an autism, then I can cure autism. But the reality is the relationship between the genes and the nerve cells is not straightforward. And the best thing to illustrate is that the human has only 10,000 more genes than this little worm, okay? And this little worm has only 300 nerve cells, and we have 10 to the 11. So you can imagine that with these 10,000 more genes, it's very difficult to understand why a human is so different than this little worm, okay? So what it tells us also is that the neural mechanism and the individual differences that characterizes us cannot all be determined by genes. And that's where the importance comes for environment and the healthy upbringing. And in many cases, we have to target this as opposed to the genes, okay? So this is another lesson that I want to give you now. And again, I want to use ADHD this time as an example, not epilepsy, but ADHD, because it's such a huge problem. So we have, in 98 to 2000, we had 9.9, so roughly 10% of the boys affected with ADHD. In 2010, we had already 12%. And when we look at the girls, they're much less affected. So 3.6 went up to 5.5. So this is kind of interesting. Why are boys more affected than girls? And also, what is causing this rapid increase? And we find that over the last decade, there's a 32% increase in the incidence of ADHD. So how do you explain this? And the largest jump, and this is kind of interesting, is it happens in the socially weak families. And there you found a 50% increase. And when you look at the heritability, so the genetic basis of ADHD, it has not changed. So these are studies from 1973, 71, up to the 2003, and they all find around 75% heritability in families, okay? So the genetic basis has not changed, but something else has changed that causes this. And there's a lot of research done, and we know already that many genes are involved in ADHD, some that have to do with dopamine, others with noradrenaline, serotonin, and these are all involved in brain regulation. So we have some genes that are correlating with ADHD, but the major lesson we learned is that these are many genes, but they have little effect. So they only cause a predisposition. Okay, so if you have a mutation in this gene, it doesn't mean necessarily you have ADHD, but it makes you vulnerable, more vulnerable to get ADHD. And really what we have to understand is that these genetic predisposition, together with negative environmental factors, that causes the ADHD. And that's the case for many disorders, okay? So it's you have a genetic mutation, but it doesn't actually mean you get epilepsy. But together with this, it can lead to this, okay? So again, coming back to epilepsy, this girl, if she wouldn't have gotten the meningitis, she probably wouldn't have gotten the ADHD. But because she had the predisposition, together with the ADHD, with the with a genetic predisposition got epilepsy. And then we know a lot about the environmental factors that causes like psychosocial stress, low social class, a stressed mother, parental conflicts, large families, birth problems, prenatal exposure to nicotine or alcohol. All these are bad factors that actually, together with the genes, then lead to ADHD. And then we know that ADHD is just one part of a symptom. So we have many other genetically correlated disorders. So ADHD is very frequent in families where you have also Tourette syndrome. And then also ADHD is very related to obsessive compulsive disorders, or also called OCD. We know that there are many families where you have some children with Tourette, others with obsessive compulsive disorders, and others with ADHD. So it's all coming together. And in fact, this is shown here. So if you have Tourette syndrome, the likelihood that you have also ADHD is very high. That you have anxiety problems, that you have depression and other problems is very likely. So it's a host of things that come together in many cases. And so it's a multifactorial disorder. Now, thinking about cures, it's very difficult to start producing gene manipulation, and you actually don't know whether it actually will help, because, as I told you, the gene is not causing the disease, it's just a predisposition. So the first hope for many of the cures is actually affecting the environment. Can we change the environment? Can we understand the environment? And can we target the environment? And so when you think about it, a lot of those environmental factors haven't really changed. So large families, if anything, the families got smaller now. We have also a much better, let's say, social system, I think. So low social class, maybe it's not enough to explain why it really dramatically increased. And what we think is happening as part of this explanation is that the media has increased, okay? The social use of media. If I talk to my children, yes, many of them, have their laptop, and then they always watch. They like, if one of my daughters was doing homework, she was doing like emails, she was Googling, she was pinging, and whatever, like all the things going on at the same time. And this is something that has changed very dramatically over the past 10 years. And there are many correlations with this and ADHD. We know, for example, that the media use is increasing with age. So if you look at the 3-month-old children, 40% of the children have exposure to TV. By the time they're 2 years, almost 90% of them have exposure to this. And then there's work done by Dimitri Christakis, also at Seattle Children's, who showed that each hour a baby watches a DVD of Baby Einstein, they actually speak 6 to 7 less words. And he got sued for that from the Disney Channel. But the facts are facts. So he found the correlation that actually what we think is good for the child, to put it in front of the TV, is actually maybe not so good. And the media use also increases with age. So by the time they're 1.5 years old, a lot of children already watch more than 1 hour. But by the time they're 3.5 years, most of the children actually watch more than 2 hours per day. And then the amount of TV they watch is actually correlated also to education. And so if you look at the high school people and compare it with those that have parents without high school degree, their media use is 37% higher than those with high school degree. And the more college you have, the less probability you have to actually have children that are totally sitting in front of the TV. And then there's another correlation that shows that boys, when they watch a TV, they watch a violent movie, become very aggressive, and much more aggressive than girls. And you say, okay, maybe it's the violence of the movie, but indeed, any movie they watch, they become more aggressive than girls. And again, this correlates with a more profound correlation of ADHD with boys versus girls. And all this are correlations, okay? So do children get overstimulated? And could that actually contribute to these problems that we're having these days? And Dimitri Christakis, when he did all this research over the past 10, 20 years, he always got criticized for that because it's only correlation. You know, how can we go about this to really get at the mechanism of this? And so he came to us at the Brain Research Institute and said, hey, Nino, we need an animal model, okay? We need something to test our hypothesis. And so, so we developed an animal model. So here comes your animal models. And in fact, we hired my son who just graduated from college, a psychology major, and he was not the typical college graduate, or maybe among those that wear sunglasses. So he was like, he was like a DJ and all this stuff. So I said, you're the perfect person to develop the animal model. And what he developed is he put these mice and then exposed them to the comic channel. And then not only to the comic channel, but also stimulated them with lights and all sorts of things. And then he looked after this, what happened to these mice? What happened if they're really exposed to a lot of those stimuli? And so he tested the open field test. So this is like just a box. And a normal mouse will only run around the rim here, the corners. It will never go into the middle. And the reason they don't go into the middle is that if they go into the open field, the likelihood that they get eaten is very, very high. So a mouse would not do this. And in fact, when you measure the walking, a normal mouse is always on the rim. Rarely they hit the middle. And after this overstimulation, these mice are all over the place. And it was highly significant. So that they were hyperactive. So they were like traveling way more. They were running around like crazy. They entered all the time into the middle and spent most of the time in the center. So these mice would have been eaten in no time. And so they were much more risk-taking. Then he looked at the learning and memory. They had to find a hole. And the first day, these mice were actually doing better. They were finding it much faster. But after a while, they never learned it perfectly. They would always make a little mistake, which again is not so good. And then he looked at how they recognized novel objects. So you give them a novel object, and they explore the novel object, and that's kind of the normal reaction. And this is a normal mouse. You know, you put this novel object in. They spent most of the time exploring it, but what he found is that after overstimulation, they actually did not remember what is novel and what's old. And so this is the statistics here. They spend more time on the novel than the familiar object, but here they don't make any difference. So they don't remember. And this is the ratio, so they just couldn't tell you, is this novel or not? And then we got into this question about addiction. You know, ADHD is very highly related to addiction. We know that ADHD patients 5 to 10 times more common among adult alcoholics. Most of the smokers actually have ADHD, and we're thinking that, you know, like this is part of this picture. And so we looked at the addiction probability of these mice. And so what we do is we put the mouse into the middle here, and then there are two compartments. In one compartment, we give them cocaine. In the other one, we give them saline, which is neutral. And what we find is that after a while, these mice go exactly there where they get cocaine. And the interesting thing is these mice that were overstimulated were highly more motivated to get cocaine than the controls. So I want to skip this. So basically what I want to tell you with this is that it only happens if you expose them as children. If you do this at adult, it is not happening. So we have now a hypothesis that a lot of the problems that we're having with ADHD may be sensory overstimulation, and we have an animal model. And so now that we have this animal model, we can ask the question, where in the brain is all this happening? And we can go and show, for example, in the hippocampus that has something to do with learning and memory, that there are deficits exactly in this brain area. I need to wrap it up, so I don't want to go into all the details, but we can now dissect exactly, you know, addiction, for example, is related to an area of nucleus accumbens, and we can show, this area is also overexcitable. So now we can come up with new strategies to not just put a Ritalin or something drug on that, but have much more specific substances that can prevent our cocaine addiction.
I have a question about the Tavian, the girl that redeveloped the seizures. When you first analyzed her histology of her brain, were the cells looked normal or they were also enlarged?
You know, when we get the brain tissue is divided into different groups. So one gets the pathology department, and the other one gets the neurophysiology department. So we do the physiology, and then the pathologists do this. So we knew, that these cells were weird, and it was kind of, for me, very interesting to see that in most cases, when we looked at this, these children had an abnormality. And this is kind of interesting because often these children come and they got something, like they got an infection or something. But then you look at the brain and you see, they were predisposed, but it took this trigger to get it. But we didn't know what to do with this, and now with this collaboration of someone who studied brain malformation for many years, the whole thing comes together, So yes, we knew about it, everybody knew about it, but we didn't know how to target it, and now we can target it because we can understand it. And this is also a very important lesson because Dr. Dobbins studied for 30 years brain malformations that are extremely rare. So there's maybe 12 people in the world. They come from Italy, from Ecuador, and all this. And he gets these samples and then finds the genes. And then everybody says, why do you invest so much time for 12 people in the world? I think it's important to do this, but they say, why don't you go for the bigger problems? But now, suddenly, a quarter of the epilepsy patients have this problem. So now suddenly, even though he studied very rare diseases, it becomes relevant for a quarter of the patients. So it's a huge amount of numbers. So when you study something, you often don't know how important it will be, and that's also something very important to learn. Mm-hmm.
Yeah, hi. I was wondering, so when I think of ADD, I think of a hyperactive child. And
Yes.
you hear ADD this, ADD that all the time. But have scientists come up with an exact neurological definition of what it is?
Mm-hmm.
Is it clear? Or do you just say, this kid is hyper, so let's say they're ADD? Let's call it ADD.
This is the biggest problem is the definition. And what we're hoping, our hope is by using our kind of brain research to better define it from the neural level. Okay, if the amygdala shows these and these symptoms, then that's part of the ADHD. So maybe we can help to shape the definition, because at the moment you're totally right. A big deal is you classify that as a bipolar, but you don't know, is this a bipolar kid or not? And oh my God, yeah, it's a big problem, the definition. But I hope our research can help to better define actually the disorder. The same thing like, epilepsy. There's such a huge amount of different kind of epilepsies, but that research could better define, what do we mean with this epilepsy that this and this person has, So, did I answer you, kind of?
So it's not 100%.
Yeah, of course I didn't. Yeah.
No,
No.
not that you weren't clear. The definition for ADD.
The definition is a moving target also.
And what is the difference between ADD and ADHD? Is
Oh man. Okay. Yeah, and the one you have the hyperactive as a component, but I think we use it all the time the same way. Yeah, we use it the same way. I know that I go to Germany and they only call it ADD. And then I come here, they call it mainly ADHD. It's the same thing with Staten infant death syndrome. Some people call it SUDEP, some SUDI, some call it SID, some call it SID. There's a lot of, fighting over maybe nomenclature. Mm-hmm.
Some animals can smell disorders in humans.
Oh, yeah.
And how many of the neurological problems can they sense, or are there any?
Oh, yeah. Yeah, I read I saw a movie once about these rats that can tell you whether you have TB or not. Fascinating. I have no idea. I mean, this is something which we should explore. Absolutely. You know.
What is it they're smelling? Or
Yeah.
If it's smelling.
Yeah. You know, the thing is, how would I say this? When you look at a disease, you know, it's very easy to look at one or two symptoms, but I think a real good doctor has the sense, you know, that because it's not always the one thing. So I can tell you one example where a mother came to me and she said, you know, do you know about Tourette syndrome? And I said, of course I know about Tourette syndrome. It's a motor tics, and she almost killed me for that because she said, look, it's not just motor tics. There's this and this and these and these and these other problems because it's the whole child, you know, and that's something that we hope to address also. I mean, that you know, they are vulnerable to other things. They have maybe allergies. They have all sorts of other things because the brain controls everything. The immune system controls everything. So, yeah, we have to start like, okay, we have to think much more holistic. And I think a mouse or a rat that smells it is looking at it holistic. You know, they look at this, how the person moves, smells, etc. But at the same time, I'm a mechanism guy, so I dissect it, but I don't forget that it's just the dissection of the brain. Yeah? God, I give you kind of woolly answers, I know. Yeah.
So, with regards to the experiments that you did with the overstimulated mice,
Yeah.
was there a minimum amount? Like, when did you start seeing the dramatic results? How much overstimulation did that take? And for that matter, was there a maximum amount of overstimulation where those results stopped getting worse?
Yeah. Like we asked for NIH money to get this question addressed. It takes a lot of work to get to this. So we started by taking these mice out from the mother. So we said, okay, let's take these little pups and expose them for like 2 hours a day. You know, is this causing something? The problem is the separation from the mother was so dramatic that we see a lot of effects, but it was because we took them away from the mother. So we had to leave them there. And then we tried 6 to 8 hours, and we got that for like 2-3 weeks. 2-3 weeks for 6 to 8 hours exposure to this. And then the effect lasted for the rest of their lives. Okay, so it was irreversible. These mice, we could test like half a year later, and they would still show these symptoms. But then we did the same thing by exposing adults, and they didn't show these symptoms whatsoever. So it was really like this critical period when they grow up, when this exposure changed the brain, and it didn't go back. So that was for us shocking. Yeah. Yeah? But I mean, we didn't titrate. I don't know exactly whether 2 hours would have been enough.
For the epilepsy patients, is there any common brain regions that can cause the seizures? Like,
Oh, yeah.
do you expect some spots?
Yeah. You know, in adults, a lot of the seizures are temporal lobe seizures, which means they start with the hippocampus, the one I showed you as a figure. In children, however, it can occur everywhere in the neocortex, and it's a lot of them are neocortex seizures, and the reason for this, I think, is that the mechanism that lead to epilepsy in a child are different than the ones that lead to epilepsy in an adult, and you have kind of like a huge peak of occurrence of epilepsies in children, and then you have kind of a period of silence, and then you gain with 40, 50. You get a lot of epilepsy in adults, and the mechanisms are very different. And I think a lot of the epilepsies that you get in children are due to kind of developmental problems. You know, it's the same thing with cancers. A lot of the cancers that you see in children are developmental cancers. Then later on, you get cancers because of, you know, let's say you smoked the whole life or something like that. So the same thing is for epilepsy. And the epilepsies that occur in adults is much more hippocampal. What's kind of fascinating to me is, you know, you see these operations and you think, oh my God, this child, you take piece of the brain out, it will be totally damaged. And the fascinating thing is that you can almost take like half of the brain out of these children and they recover and they can walk and they can go to school and etc. So that's why, you know, these operations in children are actually, you do a good thing to them even though it looks pretty brutal to take out a piece of the brain. But often, you know, the neighboring piece of brain has already reorganized, and even if you take out an area that is supposedly important for memory, the memory becomes better if you take it out. So, yeah.
Hi, I was just wondering, it sounds like a lot of interesting assays were performed using live tissue that was taken in the ER. And I was just curious to know if any live cell imaging microscopy was used to watch the real-time changes in the cell cycle, particularly in the presence of some of the drug therapies.
Oh, yes, absolutely. You know, we have a lot of microscopes, and we can actually look in the... So we do different ways. So like, for example, we have a $1 million multiphoton microscope where we can put a piece of mutated brain or diseased brain in one microscope and then a healthy brain in the other one. And then we can watch them real time, how this one is changing and this one not. Or we can put markers off for progenitor cells, and then we can observe them exactly how they grow in and how they grow in wrong in a diseased brain. So we have these things. We have other microscopes where you can train the mouse to go under the microscope, and then you can watch real life, you know, brain activity in the brain. So there's a lot of new, very exciting development, and we have incubators where you can put a piece of tissue in there. So we're studying brain tumors, for example, you know, how they invade the brain, and then you can watch that in a microscope over 4 weeks or something like this. So yes, the technology is just incredible, and you can combine it with electrophysiology, you can combine it with different environments, you can, you know, all sorts of things. So yeah, absolutely. Mm-hmm.
Sorry, just out of curiosity,
Yeah.
was one Delta Vision?
Sorry?
The Delta Vision, was that one of the microscopes that was utilized? It's an inverted.
The Delta Vision. Oh my God, no. You know, we use Olympus and Zeiss. Yeah, and I have to tell you, like, we love the Zeiss. It's totally user friendly, but costs like a Mercedes. You know, so the Olympus didn't cost that much, but you need an expert for that. You know, so yeah.
Yeah, so I was interested in you almost have two hits, so maybe you have like a genetic predisposition, you said,
Yes.
and then there was an environmental impact
Absolutely.
on that. So I'm curious if there are individuals that don't require that environmental interaction,
Oh yes.
and if that can lead you or leads to perhaps the pathway that's actually involved for the environmental stimulus.
Ah, okay. Okay.
Or how do you get at the environmental stimulus? What the pathways are in that?
Yeah.
Is another way of asking that.
Exactly. You know, okay. I think it's not black and white. Okay, so there are certain diseases where one gene causes a neurological disorder. Like one is this girl with MECP2 mutation. She has an MECP2 mutation, and that causes Rett syndrome. This mutation is sitting on the X chromosome. And if you are a girl, you have two X's and one gets always inactivated. And so depending on how much inactivation you have a full-blown symptoms or you have less symptoms. So then when it comes to ADHD, there are also different genes, and depending on which one, it has a bigger impact and environment has less impact. So it's very, very different for every disease. You know, it's not like I remember, like Kathy Milne, she got a baby, and all these risk factors, she looked at the genes that she's studying, her baby had also, and she goes, "Oh my God!" And then the baby was totally fine. Okay, so it doesn't mean, but you're right. The question is now, what are these environmental factors doing to the brain? And that's what we do. I mean, like as I said, the nucleus accumbens get totally overstimulated, get totally hyperactive. All these sensory pathways become very, very active. So we're thinking that in the disease state, these pathways are totally upregulated, that now if you give a genetic mutation to that, it brings it just over the top. Yeah? Did I understand? Not quite? A little bit. It's brain, hey, it's complicated.
I know somebody that was diagnosed with CAPD, Central Auditory Processing Disorder, and also ADD without mention of hyperactivity.
Yeah.
I know there's a lot of gray area there, and they cross,
Oh, yes,
a
exactly.
lot. He was also told that he wouldn't be able to really improve his processing speed, but he didn't buy it. So
Yeah.
he's been working hard on nutritional therapies, which was another
Absolutely.
question that I had.
Oh,
Do you do any research with regards to nutritional therapies?
Yeah, for example, we have a whole group studying like the effect of high fructose diet on the brain. That's part, but you're totally right. I mean, all your nutritional substances act everywhere in the brain. They're like neurotransmitters, and they can change it. And so, a good diet absolutely is very, very important for the health. And I had a slide prepared for that, but then I took it out. But absolutely, and then also, these predictions is so hard to take. And I also believe that every disorder is also an opportunity. Like I think. These risk-taking mice, yeah, they can get eaten, but they also discover something that the other mouse wouldn't do, and I would like to take my own personal example, I was really terrible in school. I was classical kind of ADHD, and my mother had to go to 30 high schools to find a school that took me finally, but I think that kind of ability helps me now for my science, And maybe my science is still ADHD-ish because I'm studying not only epilepsy, but I study like 10,000 different other things. So, but I think it's an opportunity, and I think the trick is often to say, how can we channel your disorder into the right way and make a strength out of it, And the temporal processing, in fact, is something that is not fixed. Like there's this Aerosmith program where they train this temporal processing. Can they speed it up? And it seems to help dramatically. And they do. They learn how to read the clock, for example, and become better and better. And as soon as they can do this, suddenly also their social interaction get better. So, a lot of the disorders we have is temporal processing, and that you can treat. And I'm thinking sometimes that our brain research is not necessarily there to develop a new drug that helps you, but maybe to understand the mechanism and ways to bring a disorder into the right direction, something like that. Yeah. So I think it's good to know that for example, your friend or something that, proved them wrong. The prediction, you're not fixed. The brain is never fixed. Yeah. Mm-hmm. But in the wrong environment, that person would have probably suffered very much. That's another thing. Without my mother, I would still probably be whatever. I don't want to know. Yeah.
So I was wondering if you could comment a little bit more. What struck me is thinking about, okay, if you're looking at pathways, and I'll say, I appreciate you putting up that signaling pathway slide, because our students are getting bombarded with all these kinases and everything right now, so I hope they appreciate that. But but if you're identifying, let's say, parts of those signaling pathways that are malfunctioning in these cell types, what struck me, and this is what I'm hoping you can give me a greater feel for, is if you think about cancer biology and similar type thing, if you find defects in signaling pathways, a lot of the advancers in cancer therapies have been on the ability to target more specifically therapies to cancer cells, which are physiologically, morphologically very different. Whereas it strikes me you're not seeing a lot of these differences. So how much more challenging is it to have specificity in targeting drugs to defective brain cells?
Okay, I think I have good news here. And so far as we have so much improved over the last 5 or 10 years, to target very, very specific nerve cells. And using viral vectors. Okay, so we find something that characterizes these hippocampus cells, and we can target a virus that goes only to these cells, and then inserts a gene, and then can heal exactly those cells. So that's what we do on a daily basis now. So we don't do anymore too much pharmacology. We manipulate certain cells so they light up, or they can be activated with light. And I mean, we can right now have a mouse where we can put those viral vectors into GABAergic neurons, and I just turn a light on and the seizure stops. So we can do so many things now in mice. And it's extremely, extremely specific. And I think the hope is that we can bring all that knowledge to the human, and then also turn on the light, and then the epilepsy is gone. You know, I mean, so we can do it already with mice. We cannot do it with human, and the reason we cannot do it with human at the moment is we cannot estimate the risk of putting these viral vectors in. You know, like a mouse lives, I don't know, a year or something. A human lives 30 years. So we have no idea what these viral will do if you implant this into someone, and then what happens 10 years down the road. Does it go crazy and infect all the other cells? So the threshold to go from all the things we know in mice to the human is really more about safety. What can we risk? But there's a tremendous advance coming up. And my hope is that a neurosurgeon in 10 years or 20 years is not cutting out a piece of the brain, but rather putting in a viral vector that stops the seizures right there without taking it out. So I think there will be a lot of rethinking coming up. And at the moment, these are all very cool tools in mice, but at one point, I hope it will go to the human. And then, as I said, if I know exactly the nucleus accumbens is overexcited in an ADHD kid, maybe I can target exactly those cells, and then that kid is still the same kid, but will not go for drugs. You know, like, I don't know. It might be futuristic, but I think there's a lot of hope. So in my talk, I emphasize a lot of the challenges, and that's my daily life, the challenges, but there's also a lot of hope, yeah? And it's all biotechnology in a way, because, as I said, I mean, like at every little step, even if we look at behavior, we use a lot of transgenic mice and a lot of technology, and we can target all the pathways very specifically in, let's say, only the dopamine neurons and only the GABA neurons or only the glutamatergic neurons. So there will be a lot of efforts, success hopefully coming in the next 10 years.
I have one. I wanted to know if certain neurological conditions saw more prevalence in certain areas of the world, so geographic differences, and then whether or not those differences were genetic or environmentally caused.
Yeah, like Bill Dobbins, for example, did an expedition to the jungle of Ecuador, because there was like a whole population of people. They have a high prevalence of hydrocephalus. You know, so because he said, okay, there's such a high prevalence, so there must be a gene that causes this, and I want to understand the gene. The same thing, I mean, there are like villages full of Huntington disease, etc. So yeah, there's a lot of genetic accumulation in different areas, and we actually use this a lot to study this. You know, like there's certain brain malformation that occur only in certain areas, and that we use to find the tools, to find the mechanism. Absolutely. Mm-hmm. Yeah.
Where does this research sit in the financial pecking order?
You know, there's a lot of inequality in some of the, like for example, epilepsy research did not get that much money like Alzheimer's disease, you know, or AIDS. You know, AIDS is very high in the pecking order. Alzheimer's not. Hydrocephalus, even though it's so frequent, is very low in the pecking order to get money. And the beauty about our institute is that we live a lot from the NIH, but also a lot from philanthropy. And we have, you know, wealthy families that help us to do, let's say, hydrocephalus research or Tourette research, and that actually plays a big role in solving this, because often it takes, let's say, and this sounds a lot, but I mean, it's a $1 million gift, and then you can generate enough data to go with this to the National Institute of Health, and then you get like $10 million grants. And so our center generates at the moment around $11 million on research funding from the National Institute of Health. And a lot of this funding is kindled by the Seattle Children's Hospital and by families that are affected, etc. So I really don't want to complain too much. It's everybody complains about how hard it is to get money, but I mean, if you run a business, it's also hard. So it's hard, but I find us very rich in what we can do and the tools that we have, and I must say, Seattle Children's invested a lot of money in research right now, and I've never been in a place where we feel so good about the commitment to research, and it's rewarding because it comes up, it helps, and I think the United States has an incredible philanthropy culture, where people are proud to give if they can. And I come from Europe where this is not the same situation. And I think a lot of the research is driven by people that dedicate their life to support research, like hydrocephalus. When I came to the Children's Hospital, I learned about hydrocephalus. I was teaching hydrocephalus. It's like a plumbing problem, And then we got a gift of $750,000 to do hydrocephalus research, and it developed into something really interesting, and we learned so much about this. So I'm thinking that the people are driving this research, and we are part of that team, And I tell you, if you see these kids, you have a different responsibility, and that comes often with this interaction with these parents, and that's why. I mean, as I said, the Seattle Children's is an amazing place to work, because you have contact with the clinicians, you have contact with the parents, with the patients, and then as a researcher, you know why you do that, And if you, so I can't complain. Everybody complains because we had a big crisis in financial crisis, but hey, everybody's affected, So.
Hi, thank you. I learned so much. My brain is like on fire here.
Open the window. Yeah.
But one of the things I was interested in hearing a little bit about was the autism research. I was just wondering, has there been some revelation, something new? Because I know you didn't actually get to it, but it was something that's I really wanted to hear a little bit about.
You know what? One of the problems with autism is very similar to the problems that I mentioned in other disorders. That autism is a spectrum. It's like not one disorder, And I gave one example, and I didn't talk about it much, but in this case, the temporal processing of these children is slowed down. So if you give them two tones, they hear only the second tone because they cannot process it very fast. And these children have hard time to socially communicate or interact because for them this processing is too fast. For social integration, when you have to listen to something, you have to hear something, if it's too fast, they become very scared and they don't want to do this. And we know exactly where in the brain this temporal processing is done, and we can target this and then hopefully heal it at one point. But that affects only these kids. And then there are other kids that have problems with habituation. So if normally if you see a face for 2 minutes, you adapt to this. You're not getting like a huge brain response to that anymore. But some of these children don't have that. So each time they see a face, they get like a total reaction to that. Again, they are very scared to interact with this. So they have another problem. And so there are many, many different disorders that are all together with autism. And many of them are temporal processing disorders, and I hope that by, for example, training, you can actually get them out of this. But I think one of the big messages I was bringing you is that even though you think it's the same thing, it's not the same thing. It's like, it's like even epilepsy, that, it's this seizure activity. It's caused by many different things. The same thing is with autism, with ADHD. So these are like a description of a disorder, but it doesn't tell you necessarily about the mechanism. Yeah.
My question is there being less to cut down for medicine? Because when I was a camp counselor, this one kid who had 3 different conditions, he had on average 4 different kinds of medicines for each condition. And I just remember when he was on them right in the morning, he was so zombified. By the afternoon, he was perfectly calm and fine, but it was just ridiculous the amount of medicine that he was getting. So I'm just wondering if
this is a little bit the problem is that's when I said, like, where do you draw the line? And every doctor has different approaches. It's difficult, these psychiatric disorders to treat is so terrible because it's like every individual will respond differently, and you have, as a doctor, you interpret, okay, this is the reason, so I try this drug. But you don't know exactly, is this drug? And then they accumulate different ones. But at the same time, in many cases, the only way to help this child, and I mean, don't get me there, it's scary. Yeah, I find it scary. But at the same time, it's also the only thing you can do often, So you should talk to a psychiatrist, and they would know better. But I prefer to treat my mice, actually. Yeah.
Was there a question in the front?
Thank you. So I was wondering if you could walk us a little bit through the process of how you take research from mice to turn it into therapy with humans, and like what that requires.
Very nice question. Yes. So let's start with Bill Dobbins. Okay, Bill Dobbins is a human geneticist. So he looks at all sorts of different disorders, epilepsy, autism. He has some autism genes. So now you don't know what this gene is doing. Okay, so you find a gene that is mutated, but you don't know what it does. So now we have Kathy Millen. She can make a mouse with exactly the same mutation as this child. Okay, and so this takes her like 3-4 months, which is amazing. It's very fast. And then you have a mouse that has supposedly the same problem as this child. And now you take a behavioral person that characterizes the behavior. Is this actually the same symptom? Okay. And astonishingly, you find that. I didn't show you the movie of this child with Rett syndrome, but we can make a mouse that has exactly the same problems as this child. And so once we have that mouse, now we can take out the brain and ask the question, what's wrong in the brain? Because we couldn't have done this with a human. So now we can dissect it, and we can take out a piece of the brain, and our brain consists of lots of different engines that do different things. Like we can take out the breathing center in a 500 micrometer thick tissue, and that breathing center is breathing as if it would be in the whole animal. And it will have the same problem as this child. So now I can study this breathing problem in this little piece of tissue, and I can use immunohistochemistry, I can use electrophysiology to really dissect what is actually causing the problem. And now I have a theory. Now I try to treat that mouse. Can I fix the mouse? Can I stop the seizure? So can I stop this? And then once I have a good drug, then we have another center which is doing clinical trials, and then they can design the clinical trials that tell us how many humans we have. And so this is kind of the process. But I tell you, it's a long process, because before you get a drug that you can go, it's oh my God, it's a lot of, you have to first show the safety nets, etc. But that's the theory, yeah? But it's amazing what you can do. I mean, like, basically you can make from every human disease kind of a mouse model.
Kind of a follow-up question. The brains of a mice and the brains of a human, are they Are they very similar? Like, what kind of differences are there?
Very,
I'm assuming there's some
okay,
difference.
very important question. Extremely important, actually. The brain is kind of hierarchically organized. So, for example, the areas that control breathing are very old. Every animal has to breathe. So these areas are very, very similar. But when you come to the neocortex with a higher brain function, the human brain is very different than a mouse brain. And in fact, there are fundamental differences. And we often assume that, okay, if we can heal a mouse, we can heal a human, and then in the neocortex, there are major differences. But still, surprisingly, the disorders that happen is, for example, too much inhibition or something. And that will also happen in a human, too much inhibition. But the consequences of this will be very different in a mouse versus human. And the Allen Brain Institute right now is using a lot of those samples to better understand what's the difference between a human and a mouse. And as I said, there are many, many differences, astonishing differences. And we're getting to learn this more and more. Mm-hmm. And that's why often you go for a monkey, unfortunately. Like sometimes you have to do primate research before you dare to go to a human, because a primate is closer to a human. But again, between a primate and a human, again, it's a huge difference. Yeah, but it's
we're really into overtime here, and I always promise these folks we're going to quit very soon. So I hope that everyone has enjoyed Dr. Ramirez's remarks, and that everybody will, with me, give him another round of applause.
Thank you.
Thank you very, very much.
Thank you so much.